Hello class, this is our first lab session and today we will learn about fungal mycelia and septa, germinating spores and fruiting bodies of the powdery mildews. One of the most used instruments in a microbiology lab is the light microscope, and this instrument uses lenses and light to magnify items up to 1000 power. And this one is very useful because it allows us to see different microorganisms. It allows us to observe, for example, an ant of 1 millimeter up to different things with a size about 1 micrometer, such as bacteria, as we can see in this picture right here. Anything below this size cannot be observed under the microscope. The microscope has different parts that we need to be familiar with in order to properly handle it. The first part that we need to know are the ocular lenses, and those are the lenses that allow you to look through the microscope to see the specimen. We also have the stage clip, which is this one right here, and it is the metal clip that holds the slide in place so we can move it back and forth, up and down, and focus it to observe the specimen we're trying to observe. We also have the diaphragm, which is right here, and it allows us to adjust the amount of light that's passing through so we can see the image in a better resolution. We can also adjust the brightness in the microscope and to increase or decrease the light amount that's passing through. And here we have the light switch, which allows us to turn on and off the microscope. To focus an image, we have the coarse adjustment and the fine adjustment. So whenever we place a slide in the microscope, the first thing we do is to bring it up or down with the coarse adjustment. And this is the larger adjustment knob, which is right here. And it brings the specimen into general focus. Once we have it right there, we can fine tune the focus and increase the detail we're observing, with which we are observing the image using the fine adjustment. When we move the coarse focus, we make like larger movements, but when we move the fine adjustment, we just move this knob very slightly to make sure we don't lose the general focus that we already have. And right here we have the objective lenses. These objective lenses have different magnification and they usually go from 4x up to 100x. The 4x is the scanning lens, and this one is the first one we should use whenever we're trying to get focus of the image. Once we have focus with the 4x lens, we can increase the power to the 10x lens. Then we can go up to the 40x lens. If we want to use the 100x lens, we will need to use the immersion oil. That's why this lens is also known as the immersion oil lens. But even though we have different magnification in each lens, the finite magnification at which we are observing our microorganism is not the magnification that we have in the lens, because the power eyepiece also has a magnification. Usually it is 10x. So to know the finite magnification we're observing our specimen, we just need to multiply the eyepiece magnification for the scanning lens, for the lens magnification. In this case, for example, if we have an eyepiece of 10x and we're using the scanning lens, which is 4x, its magnification, we just say 10 per 4, it's 40. So the magnification we're observing the specimen is 40x. It means that we are observing the microorganisms 40 times larger than it actually is. If we are using the 10x lens, then we multiply the 10x per the 10x in the eyepiece, and we have 100 magnification. If we're using the 40x lens, we say 40 per 10, it's 400, and that's the way we have the final magnification. So you can learn what we're doing. Please calculate the magnification of you are observing a microorganism if you are using an eyepiece of 15x and the mid power lens, which is 40x. So for the lab, what we're going to do next is we're going to watch the video how to use and care the microscope, which is in this link right here. We're going to navigate as well the microscope simulator, and we're going to answer the questions about the microscope on the lab quiz, which is posted on eClass. The other important technique we usually use to observe different microorganisms is the squash mount, and it allows us to prepare the slides to observe the microorganism under the microscope. To prepare the slide, we usually use dyes to color the cells and make them easier to observe. In our case, we will be using mostly lactofuchsin, and the general way we prepare the slide is we place a drop of lactofuchsin on the slide. We just get a small piece of the culture and of the fungus or the bacteria, and we place it into the dye. We pull apart the material, and we cover the material with a cover slip. I know this is not very clear when you say it in words. That's why I'm inviting you to watch the video, which is posted on eClass, and it shows step by step the procedure you need to follow to get a squash mount. When you prepare a slide to observe fungus, you will be able to observe different structures. And in fungus, we have different structural components. The basic one are the hypha, like this one, which is a mass of branched tubular filaments which are enclosed by a rigid cell wall. A mass of hypha forms a mycelium, like this one we have in the photograph, and the definition of mycelium is a mass of hypha that forms the fungus vegetative part, the thallus. And if we have fungus like a mushroom, let's say, this mass of hypha is the one that's going to compound the mushroom itself, the thallus of it. In these photographs, we have a culture of trichoderma, and here this is what you can see, the thallus of the fungus. And when you just take a piece of it and prepare a squash mount, you will be able to observe the hypha of trichoderma, and a large amount of different hypha compound the mycelium. So here we have an hypha, here we have the mycelium, which are multiple hyphas. The hyphas are divided into different cells by septa, which are the internal cross walls of an hypha. However, not all the hyphas have septa. Some of them don't have it, as the one that we have here in your right. These kind of hypha are commonly known as cenocytic hyphas, and when they have septa, they're known as septate hyphas. And they have different parts. In a septate hypha, we have different nucleus, like these ones which are in purple in this diagram. We have the cell wall, which is the wall that encloses the hypha. And within the septa, we have the pores. The singular of septa is septum. While in a cenocytic or aseptate hypha, we only have our cell wall, which surrounds the hyphas, and different nuclei. But there's no division, like physical division by the septa. One of the fungus you will be observing today is Phycomyces blackiscellianus, and as you can see in this photograph, it has slender and branched hypha, and they are aseptate. You cannot see any septus over here, so it allows us to say that it's an aseptate hypha, and those hyphas are the ones that compounds the mycelium. And another important feature of this fungus is that it usually has some refractory flat droplets inside, which are these ones that we can see in this photograph. This is a photograph which was taken using a squash mound, and the dye we used was lactoflushane as well. This is another fungus, and it causes diseases in many plants, which is Rhizoctonia solani. And as we can see here in this photograph, the hypha are usually right angle, they are branched, and they are septate. As we can see right here, these are the septa, one here, two here, three here. This is our hypha. In this photograph, we didn't use any dye. That's the reason why it looks in its natural color. And naturally, Rhizoctonia has pigmented hypha. And why are they pigmented? Because they have melanin, which protect the hypha from the microbial lysis and the UV components of the solar radiation. We can see here as well that they are right angle and that they are septate hypha. One of the important features of septated fungi is that the septus are perforated and have a pore, a central pore that allows the flow of the organelles and the cytoplasm from one cell to another. But why do hyphas, septus have those pores? They have it because this is the first line of defense when part of the hypha is damaged. For example, if I have part of the hypha damaged right here, we have a structure which is called the Wernicke body, and this one moves through the septum and it blocks the flow of the microorganisms. It becomes a plug and separate the damaged cytoplasms from the healthy cells, and impedes that the damage continues to all the hypha. Now we're going to study the powdery mildews, and the fungi that causes this disease belong to the phylum Hyscomycota. These are one of the most common, conspicuous, widespread, and easily recognizable plant diseases, and they are usually easy to observe in the upper side of the leaves. How do we see this? Here we have some photographs of the typical symptoms of powdery mildew. Which are the symptoms? Are usually spots of patches of white or grayish powdery mildew growth. They usually begin on young plant tissues. Here we have powdery mildew in wheat. This disease is caused by Erysippus graminis, and here we can see the powder on the upper surface of the leaf. There's also powdery mildew in grape caused by Uncinula nicator. We also have it in hazelnut caused by Phylactinia gutata, and in oak right here, which is caused by Microsphaera olfactoides. If you go and walk to the river valley during the summer, you will observe these kind of symptoms in many plants around the vegetation. So you can go and look by yourself, and if you have your own garden, it usually is easy to have this disease. For example, in your zucchini, or if you have tomatoes as well. So look around and you'll find it. As the seasons goes by, the fungus that causes the powdery mildew are able to form the reproductive structures, which are known as cleistothecia, and they can be observed as tiny pinhead sizes, spherical, which at first are white, as we can see in this photograph. Then they become yellow-brown as right here, and finally they become black. And these are the fruiting structures of the powdery mildew. How does it happen? So, at the very beginning of the season, after when springs began, we have the Cleistothecia which were overwintering in our diseased leaves. This Cleistothecia contains the ascus, which contain the ascospores, and they break once the tissue is viable, and the ascospores are released. These ascospores go into healthy tissue and infect the plant. And then we have disease leaf. But the disease leaf not only produce Cleistothecia, there we also have a sexual reproduction through conidia right here. So this conidia can also infect healthy tissue and cause a secondary infection and reproduce the disease. It's important to know that the disease is caused by an obligate fungus. What's an obligate fungus? An obligate fungus is that one that we cannot grow in any artificial media. So we cannot take an infected leaf with powdery mildew, bring it to the lab, and grow a plate with this one. It only grows if there is a susceptible host it can infect. This is also an interesting disease because the mycelium of this fungus only grows on the surface of the plant tissue, and it produces small conidiophores. The conidiophores are the ones right here which produce the asexual spores. Asexual spores, conidia, and sexual spores are the ascospores. That's something you need to remember. Once we collect these pine head structures that we observed, we can see under the microscope then, and this is what we have. We have the Cleistothecium, which is the brown structure, and when we break it, inside of it we can find the side-like ascus, which contains ascospores. They comes in numbers of 8. In your guide, you also have a link that guides you to a YouTube video where you can see how we collect this Cleistothecia and we observe them out under the microscope. I invite you to go and watch this video as well. It will be very useful for you to understand how we make it. As we learned before, the fruiting bodies of the powdery mildew are the Cleistothecia, and different Cleistothecia from different species have different appendage types. And the appendage is very important for these fungi because they play a hook role and help the Cleistothecium to attach to the host and to cause the initial infection. And for example, in this picture we have different genus, and we can see how Erysippe and Sphaerotheca have mesozoid appendage. Genus such as Microsphaera and Potosphaera have dichotomously branched appendage tips. Genus such as Phylactinia have bulbous appendage tips, and Uncinula has cercinoid, like hooked appendage tips. These are very important features to distinguish which is the genus we're working with. In this photograph, we have a Cleistothecia from maybe Microsphaera, which has dichotomously branched appendage tips, and here we have a Cleistothecia which likely belongs to Uncinula, and that's why it has cercinoid hooked appendage tips. So now we're going to go to a few genus of fungi causing powdery mildew, and we'll see which are the most important features of them. Here we have Erysippe, and their Cleistothecium has the aci. Usually there are several aci inside of each Cleistothecium, And the appendages are simple, they're like hypha, and they're known as myceloid. Uncinula also contains different acid, and the most important feature of this one is that their appendages are coiled or hooked at the tips, as we can see in these photographs. Phylactinia, also inside of each one of the Cleistocesium, we have different acid, and the appendages of this genus are straight with a bulbous base, as we can observe in this photograph. Microsphaera contains as well different acid, as we can observe in this photograph. The appendages are dichotomously branched. Now we have gone through most of the material you need to know to understand the overlap, and I'm inviting you to go through the lab guide that you have also uploaded and check every single step there. It's going to be very easy to follow, and however, if you have any question, just ask them. And using all the information in the slides, in the videos, and in the lab, please go through to your lab quiz and answer all the questions. You will be able to complete your lab quiz until 1 hour before the next lab session, and just feel free to go back and forth as many times as you want. And once you have completed the lab report, don't forget to click on submit, otherwise I won't have it and won't be able to check it. And please enjoy your lab.